Hello students, welcome to zoology class. Today in continuation to my presentations on cell structure and function, and I was discussing about the various cell organelles. Today I will take up next two cell organelles, and they are number 1 ribosomes and number 2 plastids. So today we'll talk about ribosomes and plastids. So first of all I will talk about ribosomes. What are ribosomes? Ribosomes are non-membrane bound and important cytoplasmic organelles found in close association with the endoplasmic reticulum. So when I was teaching you about the endoplasmic reticulum, I told you that ribosomes are associated with the rough endoplasmic reticulum. So ribosomes are basically associated with rough endoplasmic reticulum, and they are present in the cytoplasm, and they are not bound by any membrane. It means they are non-membranous bound organelles. So these are the ribosomes. And ribosome, it is a complex molecular machine found inside the living cells which produce proteins from amino acids during the process called as protein synthesis or translation. So ribosomes, they help in protein synthesis, and the process of protein synthesis is known as translation. So during the process of translation, ribosomes helps to produce proteins from the amino acid. We know that the chain of amino acids, they form the proteins, and the process of protein synthesis, it's the primary function which is performed by all the living cells. So this is a basic function of the cell to produce proteins for the living cell. So ribosomes, they are found in the form of the tiny particles in a large number of cells, and they are mainly composed of 2/3 of RNA and 1/3 of proteins. And ribosomes are the most important cell organelles composed of RNA and protein that convert genetic code into chains of amino acids. So what is the composition of the ribosome? Ribosomes are made up of two things, RNA and protein. It is composed of 2/3 part is RNA and 1/3 part is protein. And these ribosomes, they convert the genetic code into chains of amino acids, and these amino acids, they form the protein. So this is the basic function of the ribosome, that is to produce proteins. So each ribosome, when we talk about the structure of the ribosome, then each ribosome, it consists of two subunits, the large subunit and the small subunit. And the large subunit, it is generally dome shape, and the small subunit, it is oblate, that is the cap shape. So this is the structure of the two subunits of the ribosome. And what about the size of the ribosome? The size of the ribosome, it is determined by the speed with which they sediment in a centrifugal field. And Swedberg unit, that is, it is represented by S. This is the unit to measure the sedimentation speed. So when we talk about the size of the ribosome, then we determine the size of the ribosome by the speed with which it gets sedimented in the centrifugal field. So sedimentation speed in the centrifugal field, it is the size of the ribosome. And the unit used for the size of the ribosome is represented by S, and it is the Swedberg unit, which is used to measure the sedimentation speed, and that is the size of the ribosome. And they are known as 70S, that is the size of the ribosome can be 70S or 80S. When we talk about the prokaryotes, then the size of the ribosome is 70S. And when we talk about the eukaryotes, then the size of the ribosome is 80S. And When in prokaryotes it is 70s, then the size of the larger subunit it is 50s and the smaller subunit is 30s. It means the 70s ribosome in prokaryotes, it has a small subunit that is having the size of 30s. It means it sediments at a speed of 30s in the centrifugal field, and the bigger subunit it sediments at a speed of 50s in the centrifugal field. So this is not necessary that 30 plus 50 should be 80. No, this is the sedimentation speed. And in the same way, in case of eukaryotes, when ribosome it is of 80s size, then its bigger subunit, that is the larger subunit, is 60s and the smaller subunit is 40s. So this is the size of the subunits in prokaryotes and eukaryotes, and both 70s and 80s ribosomes, it means they are composed of two subunits. One is larger subunit and other is the smaller subunit. And ribosomes are either encompassed within the endoplasmic reticulum or they are freely traced in the cell's cytoplasm. So ribosomes are either present within the endoplasmic reticulum, that is a rough endoplasmic reticulum, or they are present freely in the cytoplasm of the cell. And the ribosomal RNA and ribosomal proteins, these are the two components which form the ribosomes. It means ribosomes are made up of two things. One is RNA and other is the protein, and these help in protein synthesis. So this is the ribosome. So this is the diagram showing the presence of ribosome. So this is the cell and this is the nucleus and this is endoplasmic reticulum, and on these you see small dots. These represent ribosome. So this is the location of the ribosome in the cell. So if we talk about the subunits, then in case, as I told you, that in prokaryotes ribosome is 70s. So this is a 70s ribosome and it has two subunits. Bigger one is 50s and smaller one is 30s. And in case of eukaryotes, it is 80s ribosome and its two subunits are these. That is the bigger or the larger subunit is 60s and the smaller subunit is 40s. So these are the subunits in prokaryotes and eukaryotes. Then we come to the function of the ribosomes. So what is the main function of the ribosome? So the main function or the primary function of the ribosome is protein synthesis in all the living cells, which ensure the survival of the cells, because if proteins are present, only then a living cell can survive. And the important function of ribosome is to synthesize proteins, and then it assembles amino acids to form the proteins that are essential to carry out cellular functions. So you know that what are proteins? Proteins are basically the chains of the amino acids, and ribosome, it assembles the amino acids to form the proteins to carry out the various functions of the cell. And the proteins synthesized in the cytoplasm, they are utilized in the cytoplasm itself, and the proteins synthesized by the ribosomes, they are transported outside the cell. It means the proteins which are synthesized by the cytoplasm, in the cytoplasm, they are used in the cytoplasm only, but the ribosomal proteins, that is the proteins which are synthesized by ribosomes, they are transported outside the cell also. So these are the functions of the ribosomes. Next comes the second organelle for the today's topic, that is the plastids. And what are plastids? Plastids are double membrane organelle which are found in the cells of the plants and algae and some unicellular organisms like Euglena. And so plastids are responsible for manufacturing and storing of food, and these often contain pigments which are used in the photosynthesis, and different types of pigments, they can change the color of the cell. So plastids are basically are the double membrane organelle present in the cytoplasm of the cell, and these plastids, they are present in the plant cells, algae, and some unicellular organisms like Euglena. And plastids, they bear some pigments, and depending upon the pigment, the color of the plant is present. So they impart color to the plant. And what is the function of the plastid? Plastids are responsible for the manufacturing and the storing of the food. That is, these pigments present in the plastids, they are used in the process of the photosynthesis. So this is the main topic or the main function of the plastids. Then what are the types of the plastids? So there are different types of plastids with their specialized functions, and among which few are mainly classified based on the presence or absence of the biological pigment and their stage of development. So when we talk about the types of the plastids, there are different types of plastids and they have specialized function, and depending on the presence or absence of a particular pigment, they are named. So there are many types of plastids, and the main types of plastids, they are chloroplasts, then chromoplasts, then gerontoplasts, and leucoplasts. So these are the four main types of the plastids, and chloroplast is the basic type of the plastid. So this is the main, chloroplast is the main type of the plastid. Then first of all, I will take up chloroplast. Chloroplasts contain chlorophyll, which imparts green color to the plants, and carotenoid pigment is present in chloroplast, which are responsible for trapping the light energy essential for photosynthesis. It means that during the process of photosynthesis, chloroplast containing chlorophyll, it is responsible for trapping of the light energy from the sun. Then second comes the chromoplast. Chromoplasts contain fat soluble carotenoid pigments like carotene, xanthophylls, etc., which impart yellow, orange, or red color. So chromoplasts are basically carotenoid pigments like carotene or xanthophylls, and because of these pigments, they impart yellow or orange or red color to the plant. And then the third type of plastid is the gerontoplast. So basically these gerontoplasts, they are basically chloroplasts only that go with the aging process. After the aging process, chloroplasts become gerontoplasts, and gerontoplasts refer to the chloroplasts of the leaves that help to convert into different other organelles when the leaf is no longer using photosynthesis, usually in the autumn month. It means when leaves are not undergoing the process of photosynthesis in the autumn months, when the chloroplast of the leaves, it becomes gerontoplast. So these are the chloroplast, then chromoplast and gerontoplast. Next comes the leucoplast. So what are leucoplasts? These are non-pigmented organelles, that is, they are colorless, and leucoplasts are usually found in non-photosynthetic parts of the plants like roots, and they act as a storage shed for starch, lipid, and proteins depending upon the need of the plant, and they are mostly used for converting amino acids and fatty acids. And leucoplasts, they are of three types. It means that leucoplasts have no pigment, and so they are colorless, and leucoplasts are present in those parts of the plant where there is no photosynthesis, for example roots, and they uh, play role in the storage of the starch, lipids and proteins depending on the need of the plants. And the three types of leucoplasts are: number one, amyloplasts. Amyloplasts are greatest among all the three, and they store and synthesize starch. So this is first type of the leucoplast. And the second type of leucoplasts are proteinoplasts. These help in storing the proteins that a plant needs and can be typically found in the seeds, because proteins are present in the seed portion of the plant. And the third type of leucoplasts, they are the elaioplasts. So elaioplasts, they help in storing fats and oils which are needed by the plant. So these are the three types of the leucoplasts. So we have basically four types of plastids: chloroplast, chromoplast, gerontoplast and leucoplast. And leucoplasts are of three types: amyloplast, proteinoplast and elaioplast. Next, I told you that chloroplast is the main type of the plastid. Then we will study the structure of the chloroplast, that is the general structure of the chloroplast. So generally chloroplasts have a spherical shape, that is oval shape, which may be as a result of being pressed against the cell wall by the large vacuole in plant cells. However, this shape may vary depending upon the location of the plastid. So chloroplasts are basically spherical or oval because they are pressed against the cell wall, because the plant cells have a large vacuole, but however their shape can vary depending upon the location of the plastid. So the morphology has also shown to be dynamic, that is, it is variable, which means that the general shape can change over time. And studies have shown the plastid to be polarized, and it ranges from 5 to 10 micrometers in width depending upon the plant. So this is the size of the chloroplast. And the chloroplast, its basic structure has a double membrane envelope consisting of an outer membrane and the inner membrane, and these are the phospholipid layers. Then the space within the double membrane, it is covered with an aqueous matrix, and that matrix is known as stroma. And this aqueous matrix, it contains many enzymes and proteins which are essential for the various processes of the cell. And other structures in the chloroplast inside of the two membranes, they are the grana. Grana are the thylakoids arranged in stacks, that is one on the top of the other. Then peripheral reticulum, that is the membranous tubules arising from the inner membrane. Then chloroplast DNA is present, and ribosomes are also present here. So these are the main structures which are present in the chloroplast. So this is a diagram which shows the various structures in chloroplast. So I told you that chloroplast has a double membrane. So this is the outer membrane, and this is the inner membrane, and this is the intermembrane space. And inside this is the aqueous matrix which is known as the stroma, and in the lumen this is present the stroma. This is the aqueous fluid like structure thing which is known as stroma, and these are the thylakoids. 1, 2, 3, these are the thylakoids, and when the thylakoids are arranged one above the other, they form the granum, and the plural of the granum is the grana. So actually granum is the stacks of thylakoids. When thylakoids are piled upon one another, they form the granum. And these structures which join these things, which it is known as the lamella. So this is the basic structure of the chloroplast. So next comes the function of the plastids. So I have already told you that the main function of plastid or the chloroplast, it is photosynthesis. It helps in photosynthesis, and plastids are the sites of manufacture and storage of important chemical compounds used by the cell. So they are also responsible for the manufacturing and storage of important compounds of the cell. So they are primarily responsible for activities related to making of the food and storing of the food, and they often contain pigments used in photosynthesis, and the type of pigments in a plastid determine the color of the cell. So the pigments are present in different types of the plastids, and the type of pigment present in the plastid, it determines the color of the cell. So these are the functions of the plastid. So this is all about the plastids. So today we have talked about ribosomes and plastids. So you have to make notes by consulting these presentations materials, and I have already sent you a study material, so you have to consult both of the things, and you have to prepare notes, and you have to go through these topics. And if you have any difficulty, you can note down the doubts and we can discuss it afterwards. Thank you.